right, in this video we're going to be doing a review over Unit 2 in AP Biology, Cell Structure and Function. Rather than going over each individual section of this unit, I'm just going to kind of give it a broad overview, and we'll definitely be hitting the major points of this unit. Though if you have questions, I encourage you to ask them in the comments below. Please also like and subscribe. Like this video, subscribe to my channel. I'm always trying to improve and create new videos, and so I encourage you to do that if you're interested, not only for AP Bio, but also as you continue on your career. These videos will work, and particularly for your first couple of classes in college if you're planning any sort of biology curriculum. So I encourage you to do that and share with your friends as well. So let's begin Unit 2, Cell Structure and Function, just by talking about some of the cell structures and their functions. First, we're going to look at the ribosome. Ribosome is an interesting cellular component because it is found in all living things. So that shows common ancestry amongst all living things. Ribosomes are the site of protein synthesis, and so this is where proteins are going to be made primarily. Well, not primarily, only ribosomes are going to be made here. Ribosomes are tiny little cellular particles too. See, this represents messenger RNA here. So these are individual nucleotides. And so this gives you an idea of the scale of a ribosome. It's very small. It's not like these other organelles that we're going to talk about that actually have internal spaces. This is more like a cellular particle. Still a very important component of cells. Next is an endoplasmic reticulum. Endoplasmic reticulum is more of a membrane-bound organelle. You can even see the ribosomes that are embedded on the surface of this particular endoplasmic reticulum. The endoplasmic reticulum as a whole serves as kind of a transport system throughout the cell system of membranes, but the different kinds of endoplasmic reticulum are going to have different functions. The rough endoplasmic reticulum, which is called rough because it's covered with ribosomes, is primarily going to be a spot for newly formed proteins to be folded up and then transported out of the cell, and we're going to talk about that endomembrane system as well. And the smooth ER has a number of functions. It could be membrane lipid synthesis, like phospholipids that are part of the cellular membrane, detoxification of cellular components, and there are different kinds of smooth ER located in different sorts of cells that have other functions, like muscle cell have a special smooth ER that is a place for calcium storage and disposal. So that is, depending on the type of endoplasmic reticulum, it's going to be particular to a cell. The rough ER again is going to be where the ribosomes are making the proteins. They're going to insert those proteins into this area, this lumen of the rough ER, and those proteins are then going to be shipped out of the rough ER toward another location, which we'll talk about next. This is the Golgi complex. See this here we have these incoming transport vesicles. This incoming transport vesicle has some sort of protein in it that is coming from the rough ER and is going to find its way to the Golgi complex, or the Golgi apparatus, or the Golgi body. Golgi is going to be the common word here, which is not a really common word, so it's easy to remember. The purpose of the Golgi complex is the modification of proteins. These proteins are going to come into the Golgi complex. They're going to be melded into this system of flattened sacs, and they're going to be given some sort of modification. That modification is going to be basically is going to be analogous to taking a package to the post office and then modifying your package. How do they modify it? Well, they add an address to it, or they add some sort of stamp to it, or designation that's going to say this is going here and this is how it's getting there. And so the protein is going to be modified inside the Golgi complex and then put it into a secretory vesicle. Just means secrete or get rid of. Anytime you see this word vesicle, it's usually some sort of temporary storage container, and this is going to be sent to the cell membrane so that it can be sent out of the cell. So anytime proteins are made in the ER, they're eventually going to be heading out of the cell, whereas if proteins are made in just free-floating ribosomes, they're going to usually stay in the cell. Next is everyone's favorite, the mitochondria. Mitochondria is the site of cellular respiration. Cellular respiration is taking sugars and other organic molecules, breaking them down for the production of ATP. Mitochondria are different among cellular components in that not only are they a membrane-bound organelle, but as you can see, they have two membranes. We have this outer membrane and an inner membrane that has a series of folds in it. These folds are going to allow for increased surface area to volume ratio, which increases the availability of work. You can also see here that these little dots represent ribosomes. This little curled up thing, that's DNA. Mitochondria has its own ribosomes and its own DNA, probably pointing to the fact that they were at one point their own organism. So we're going to talk about that momentarily as well. Mitochondria, the site of cellular respiration, and plant cells also have mitochondria. Just throwing it out there because the next thing, a lot of times people say mitochondria, animals, chloroplast plants, and it's not the case. Chloroplast plants, yes. Mitochondria, both. It's not chloroplast. This is lysosomes. Let's talk about lysosomes. Lysosomes are basically a site of recycling and disposal in the cell. Each of these little lysosomes contains digestive enzymes that are significantly different chemical composition from the inside of the cell. And these lysosomes then, inside of the lysosome, basically, or the lysosome itself will join with some sort of other vesicle or food particle or something, anything, bacteria, virus, whatever, and it's going to digest that. It's going to take it apart. It's going to use those parts to make more of you, whatever that is. So lysosome, recycling of old cell parts, digestion of things that, like foreign particles that may come into the cell. Vacuoles. Vacuoles are just essentially storage facilities. Plants have a large central vacuole for water storage. Other types of cells can have vacuoles as well. Could be for fat, water, carbs. There's lots of things that vacuoles can be used to store. Very simple organelle. Chloroplasts are next. Chloroplasts are similar to mitochondria in that they have a double membrane. As you can see here, they have their own ribosomes and DNA as well, but chloroplasts are different in that their surface area to volume is accomplished by these little discs called thylakoids. These thylakoids are the site of photosynthesis. So chloroplast's main function is to perform photosynthesis, both reactions of photosynthesis, which we'll get into in another unit. So chloroplasts, very similar to mitochondria in that they have that double membrane, own ribosome, DNA sort of thing, but definitely different function, and these are found not only in plant cells, but in some other eukaryotic cells as well. Let's talk about surface area to volume ratio. Surface area to volume ratio will determine the efficiency of a cell. The higher the surface area to volume ratio, the more efficient a cell will be. So let's use this picture here. Surface area is a simple equation for a cube. It's just the number of sides, the area of one side times the number of sides. So if you have a cube that has a side of 1, then the area is going to be 1, and there's 6 sides, so the surface area is going to be 6. The volume is 1 times 1 times 1, and so the surface area to volume ratio is 6 to 1, or just 6. Well, notice what happens to the surface area to volume ratio as the cube gets bigger. It goes way down. So what do we mean by efficiency? The best way to describe this is take, let's have 2 coolers, and in one cooler we're going to cool some drinks, and in another cooler we're going to cool some drinks, but in one cooler we have little cubes of ice. Let's say we have 2 pounds of little cubes, and then the other one we have a 2-pound block of ice. Which one is going to cool those drinks more efficiently? It's the little ones. We know this inherently. The reason this happens is because more ice is able to surround the cans or whatever you have your beverage in, and is able to cool those down more efficiently. More ice is touching the workspace, right, as opposed to the one cube. And so what this is showing you here is that if you just take that same area or same volume and divide it into lots of little cells, you're going to get so much more work done. Like literally 6 times the work that you would get done, or 5 times the work. And so surface area to volume ratio, higher the ratio, more efficient the cell is going to be. This is going to work itself out in lots of ways. Cells will be modified in order to account for this. Root hairs on plants are a great example of this. What is this? Each one of these little hairs represents the plant's ability to go out and get nutrients and water for the whole plant. If this was just no root hairs, or there was just one big root with not all the surface area, it's still going to be able to absorb things, but a whole lot less efficiently than each one of these little hairs being able to absorb. The same thing is happening in our own bodies, as our small intestines represents surface area to volume ratio. As you've been told since you were little that the small intestines can wrap around the moon 6 times, or whatever your teacher told you. None of that's really true. Though it does probably have the surface area of like a tennis court, which is still pretty impressive that all of that is shoved inside your body, because there are folds that have folds. Wait for it. No, keep waiting. That have little folds on them. And so all these little bitty folds are increasing the surface area to volume ratio of the small intestine, whose main purpose is to absorb nutrients, which is good. Now, if you're an elephant, you really appreciate this idea of surface area to volume ratio as well. You have big ears. The reason you have big ears is because you have a big body. You have a big body that's constantly generating heat via cellular respiration. If you can't get rid of that heat, you will burn to death. And so the elephant has big ears in order to decrease its surface area to volume ratio or increase its surface area to volume ratio, thus ability to get rid of more heat better. Mice are doing the opposite. They're tiny. They're so small. Their surface area to volume ratio is really high compared to the elephants. And so it's going to have trouble keeping heat in. It's going to have the exact opposite. And so what does it do in order to stay warm? It's going to huddle up, thus decreasing its surface area to volume ratio and decreasing the amount of heat loss. And so this is the exact opposite. It's fascinating way that nature works itself out that way. And the same thing can happen through nutrient exchange. Plants have these little mouths that are called stomata. And these stomata allow for CO2 to come in and water to come out. Both are necessary portions of a plant's life. And as a plant has access to CO2 and access to water and appropriate sunlight access as well, it's going to create more stomata. If it doesn't have a lot of CO2 or it needs to conserve water because water is released through the stomata, then it's going to have less of those stomata per square inch or square centimeter than will have a plant that has more ideal conditions. And so nutrient exchange, another thing that is using this idea of a square or surface area to volume ratio. And that brings us to the plasma membrane. Lots of things that we're talking about with the plasma membrane. You could actually talk about all the functions of the plasma membrane using this picture, but I'm not. I'm going to use several other pictures. This is just an introductory picture. Look, here's the plasma membrane. The plasma membrane is made up of phospholipids. These phospholipids are have a hydrophilic head. Sometimes you'll see them called amphipathic, which amphipathic just means that they have one area of them is hydrophilic, the other is hydrophobic. Hydrophilic head that has a phosphate area here, which is a negative area, so that gives it hydrophilic properties, which means that it loves water. And the hydrophobic tail, which is just two fatty acid chains, extremely hydrophobic, and they do not like water. And so when you put a bunch of these phospholipids together, they're going to organize themselves so that the heads are facing toward the water, either outside or inside the cell, and the tails are facing each other because they don't like water. And this makes this nice little boundary between the cell and the world around it. There are proteins that are able to bridge that gap. Those proteins that are able to bridge that gap are called integral proteins, and these integral proteins actually they make the outside and the inside of the cell talk to each other. It could be some sort of tube here, or it could be some other sort of structure that is going to basically allow for the communication between the inside and outside, because those proteins go all the way through the membrane, integral, all the way through. And so that means that these proteins are usually also amphipathic. And then there are peripheral proteins, meaning that they're just on the periphery or on the outside, either the outside or the inside layer of the cell. These are going to have different functions depending on what types of proteins they are. Let's look at those functions. There are several functions that membrane proteins can have. They can be transport. So this is literally transporting materials in and or out of the cell. They can have an enzymatic function. Enzymes lower the activation energy and thus speed up chemical reactions. They can have a signaling function, whereas the protein represents a signal, like kind of like a door, like you have to knock here first before I'll let you in sort of thing. They could have cell recognition. Hey, this is us, let's be nice to it, or hey, that's not us, time to kill it, sort of thing. They can have an intercellular joining, literally stitching cells together so that they don't fall apart. And attachment to the cytoskeleton extracellular matrix. Basically the cytoskeleton inside the cell represents the support structure for the inside. The extracellular matrix represents the support structure for the outside of the cell, also some communication properties. And so what this allows these things to do is, again, communicate inside and outside the cell. All of this, and let's go, before we talk about fluid mosaic model, let's go back up here because this picture shows a fluid mosaic model, I think, a lot better than the picture that I chose. This shows, so you have all these different pieces here. You have the phospholipids, you have the integral proteins, you have the lipids and other things going on here. It's called a mosaic. Lots of different pieces that make up a picture, and it's called fluid because if you were to see this thing moving, I think this picture, that's the purpose of this picture. It's not currently moving because this is a different program. This little stuff would be moving around. This is not a static function. It's not static, but it's dynamic, meaning that stuff's constantly changing places. It's fluid, but it's a mosaic. And this is the fluid mosaic model is oftentimes used as a word to describe the composition of the cell membrane. One of the other things that the cell membrane is composed of is cholesterol. Cholesterol is a steroid, so it's a lipid, and it has a couple of functions. Cell membrane flexibility and fluidity is one of the main functions of that. Cholesterol can also serve as an antifreeze and for certain kinds of organisms that need that, particularly plants. And so cholesterol is also found in cell membranes. Glycoproteins and glycolipids, so proteins, lipids we know, glyco we know too because we know glycogen, which is a type of carbohydrate, and essentially these are like molecules that have two things. It's a protein that has a glyco on it, or a carbohydrate, or a lipid that has a glyco or a carbohydrate on it. And this is essentially going to be signaling type molecules for cell-to-cell recognition or any sort of hormonal function, like so cells can receive or send those sorts of things. It's just communication. And one of the things that the cell membrane is said to be is selectively permeable or semi-permeable. Sometimes you'll see. Basically what this means is the cell membrane decides what is coming in and what is going out. It is not something, it's not completely impermeable, but it's not completely permeable either. And so sometimes you'll see that semi-permeable to represent the structure of the cell membrane, deciding what goes in and out of the cell. And this shows you how those things can get in and out using types of proteins. You have different kinds of proteins. You have like protein channels, which just are basically like a straw. Now, not anything can go through there. The inside of this tunnel is going to have a particular chemical property that is usually going to invite a particular kind of chemical, whereas these carrier proteins do exactly what they say they do. They carry things through, usually using energy, whereas a protein channel is going to be a passive sort of transport. This is usually going to represent active transport or transport that requires energy, which we'll talk about in the next video. And that brings us to cell walls. Cell walls are found in plants and bacteria and fungi. In each of those types of organisms, they represent a structural component for a cell. That doesn't mean that they don't have other sorts of passive, not passive, but secondary functions, but primarily it's going to allow that cell to have structure, integrity. Like a bacteria, for instance, that cell is so small and is singular that that cell wall literally keeps it intact. Plant cell walls allow plants to grow tall and be able to reach up to the sun, support all the water that's necessary for their body processes, and so cell walls keep cells from exploding. Cell walls are also not a passage or an impenetrable barrier. Sometimes cell walls will have these little holes in them called plasmodesmata, which allows these two cells to communicate back and forth, almost like the door between two classrooms, like I have right behind me here, that leads to my favorite chemistry teacher, Mr. Krug. If you want to know anything about chemistry, you should check out his YouTube channel as well. He is quite popular. And cell walls can be made up of different kinds of components depending on the molecule or depending on the organism. If you are a plant, then your cell wall is going to be made out of cellulose, which is seen here. If you are a fungi, your cell wall is going to be made out of chitin, which is another type of carbohydrate seen here. It's going to be a lot more rigid. This is like bug exoskeletons are made out of chitin. And if you're a bacteria, your cell walls are going to be made out of this structure. And I'm sitting here trying to think of the name of this structure, and I can't do it, sadly. But that's what they're made out of. It's another type of carbohydrate here, but this carbohydrate has some proteins. So it's called proteinoglycan. I knew I'd come up with it if I just talked about it longer. Proteinoglycan is the name of the component that cell walls in bacteria are made out of. And that leads us to the final subject in this particular video, and that is the idea of compartmentalization. This is something that is in the AP curriculum that is not a hard concept, but is something that we definitely need to be talking about. Compartmentalization is the idea that cells have compartments. Why would you want a compartment? Well, the way that I explain it to my students is if I had a 1,000-piece jigsaw puzzle and I scattered the pieces throughout the school property, it would take a long time for you to put that puzzle together because there's no particular place to do the puzzle. Every place is a place to do the puzzle, and it's never going to get done. But what if we have one table that we would do that puzzle on? It's going to be happening quicker. It's going to be a designated area. And so think of compartmentalization as designating places for particular things to happen. Examples: lysosome, designated area for the breakdown and recycling of materials. If it was happening throughout the cell, the entire chemistry of the cell would have to change. It'd be just like if we ate food and it just fell somewhere inside of our bodies. It wouldn't be very efficient. It falls into our stomach. Stomach is an example of a compartmentalized area that does a particular thing. Lysosome, similar idea. Mitochondria, compartmentalization of the concept of cellular reproduction. It doesn't happen just anywhere, it happens here. And the same thing with chloroplast. Photosynthesis doesn't just happen anywhere. If it did, it would be less efficient, but it happens in these places that are made specifically for photosynthesis. That is the concept of compartmentalization. Where do mitochondria and chloroplasts come from? And compartmentalization in general. That is the concept of endosymbiosis. Endosymbiosis is the theory of how prokaryotes evolved into eukaryotes. And the idea is that some ancient cell long ago ate a smaller cell, and instead of consuming it and taking apart, they basically formed a symbiotic relationship. Symbiotic means literally live together relationship. Well, endo just means inside, living together. This thing lived inside this other one. They formed a symbiotic relationship. Now this little guy is making ATP by breaking down sugars, and this big guy is protecting it, and they have a nice relationship with one another. This is considered the origin of mitochondria. Mitochondria point to this by having their own DNA ribosomes and a double membrane. Next would be chloroplast. Same idea. Same idea as the previous one, except for that cell that has mitochondria now eats a green cell that happens to just be able to be good at photosynthesis and making the first precursors to the first plant cells. So that is part 1 of Unit 2. In part 2, I will be going over cellular transport exclusively and be looking at those concepts. Hope to see you there. Again, please like this video, subscribe, and that will help me a lot. Thank you.